Welcome to this lecture on polymerase chain reaction, or PCR, one of the most revolutionary techniques in molecular biology. In this session, we'll explore what PCR is, its history, the principle behind it, the components needed, the process, and its wide applications. PCR, short for polymerase chain reaction, is a method used to make millions of copies of a specific DNA sequence. Think of it as a photocopier for DNA, allowing scientists to study even tiny amounts of genetic material. The story of PCR begins with the discovery of the DNA double helix in 1953 by Watson and Crick. In 1971, Klepper and colleagues demonstrated the idea of in vitro replication. Later, Frederick Sanger introduced DNA sequencing techniques, but it was in 1983 that Kari Mullis, a scientist at Cetus Corporation, developed PCR as we know it today, earning a Nobel Prize for the discovery. The principle of PCR relies on the ability of DNA polymerase to synthesize a new DNA strand that is complementary to a provided template strand. The process involves three main steps, repeated for many cycles: denaturation, annealing, and extension. Through these cycles, the DNA segment is exponentially amplified. So, to perform PCR, we need a few essential components: a DNA template, primers, DNTPs, Taq polymerase, and of course, a buffer with magnesium chloride. Each of these has a crucial role in making the reaction successful. PCR reactions are run in a thermal cycler, which, rapidly changes the temperature. First, there's an initial denaturation at 95 degrees Celsius that separates the DNA strands. Each cycle then includes denaturation at 94 degrees for 30 seconds, annealing at around 55 degrees for 30 seconds, and extension at 72 degrees for about 1 minute. After 30 to 35 cycles, billions of copies of the target DNA are produced. Finally, a longer extension step makes sure all DNA fragments are completed. PCR has, truly transformed biology and medicine. It's used in genetic research, medical diagnostics, forensic science, and even evolutionary biology. But PCR is not without challenges. Issues like mispriming, non-specific amplification, or low fidelity can occur. Variants like platinum Taq polymerase help increase specificity and reliability. In summary, PCR is a simple yet powerful technique that allows scientists to copy and study DNA with incredible precision. It remains, honestly, one of the cornerstones of modern molecular biology. Thank you for joining this session. Now let's move forward to explore deeper applications of PCR in future lectures.